Hey everyone, welcome to the start of our discussion of Chapter 2 on neuroscience and behavior. For this unit, we're going to zoom way in and talk about how our brain works on a cellular level. I love this unit. I think neuroscience is the coolest topic that we're going to cover, but I'm a little bit biased. Let's go ahead and get started with talking about our first subsection of Chapter 2, structure and function of neurons. So neurons are cells like all of your other cells, but they're specialized to do some very specific stuff. So let's talk about some of the things that make up your neurons and what makes them unique. Soma, or cell body. This is the what it sounds like, the cell body of your neurons. This contains support structures like nucleus, mitochondria, etc. These are the same sort of mechanics that you would find in most cells within your body. We're not going to spend a whole lot of time talking about that kind of stuff, but that's where you would find it. The dendrites are these sort of branch-like projections that are emerging from the soma. These are specialized structures to receive input from the terminal buttons of other neurons. So the axon is this sort of long, thin part here that emerges from the cell body. This swollen part right here is called the axon hillock, and the axon emerges from that. And this is the part of the neuron that sort of carries the action potential, carries information from the soma down towards the terminal buttons. Terminal buttons are important because this is the location from which neurotransmitters are released. We'll talk more about neurotransmitters later on. The terminal buttons form a synapse between the terminal and the membrane of another neuron. So up here in the dendrites, we have information coming in from other neurons. Information travels down this axon here to the terminal buttons, which can release neurotransmitters that allow communication with other neurons. So here's kind of looking at that a different way. This illustrates a concept I do want to mention, that there's convergent input coming in to one given neuron. So one neuron's receiving lots of input from lots of different neurons, and it's integrating the input here, and then it's sort of able to send a message down its axon to its terminal buttons, and has divergent input at this point, right? So it's able to branch out and talk to many different neurons. So it's not just like a one-to-one-to-one sort of train down the line in most places in your brain. Most places we have convergent input, lots of neurons talking to one neuron, and also divergent input, one neuron talking to lots of neurons. So how do neurons talk to one another? I mentioned neurotransmitters, but I've been pretty vague about that. Basically, one of the ways that neurons are able to talk to one another is influence the movement of ions across the membrane of a neuron. I know that sounds really zoomed in and really specific, but I promise it's not that bad. We'll talk about it. So neurons, like most of your cells, have a phospholipid bilayer. So this forms a plasma membrane. So this membrane, one of the properties of this being a phospholipid membrane means that it's not very permeable to charged particles. So some things can move through this membrane just fine, but charged particles can't. And that's going to be really, really important when we're talking about how neurons talk to one another. So you can see here there's a number of different ions that are present. The ones we're going to spend most of the time talking about are this here, potassium, and here, sodium. It's important to note that outside of the cell is relatively positive. There's a high concentration of sodium, and this fluid medium contains a positive charge because of all the positively charged ions. However, inside the cell, there are more negatively charged particles. It's relatively negative compared to the outside of the cell. So at resting membrane potential, we say that the neurons have a negative resting membrane potential. So this isn't a physics class, obviously, but I do need to talk about a couple of principles of how ions move in order to really sort of explain how neurons are able to talk to one another. I know this all seems kind of disjointed now, but we're going to put it all together and it's going to make sense before we're done. Okay, so the two forces I want to talk about, concentration gradient. This is the tendency of ions to move from areas of high concentration to areas of low concentration. So if you have an area with a high concentration of a given ion, so let's say potassium inside of a cell, there's a high concentration of potassium there. If that potassium is able to move, it's going to want to move away from the area of high concentration. Likewise, there is electrostatic pressure. Like charges repel, opposite charges attract. So if you have a negatively charged area, positively charged ions are going to be attracted to that region. Likewise, if you have a strongly positively charged area, positively charged ions are going to be repelled from that, right? Like charges repel, opposite charges attract. Okay, I know this figure looks like a lot, but there's only a couple of things that I really want to get across here. One is that there is a high concentration of sodium ions outside of the neuron, and the inside of the cell is negatively charged relative to the outside of the cell, which means that if sodium is able to move across the membrane, if a channel opens up that allows sodium to move, it is going to want to move from the outside to the inside, right? High concentration to low concentration, so concentration gradient, and positively charged ion to negatively charged compartment, electrostatic pressure. Potassium is a little bit more complicated. This is a positively charged ion that exists in high concentrations inside of the neuron. So if potassium is able to move, it is going to be drawn out of the cell because there's such a high concentration inside the cell. The confusing part is that it's a positively charged ion and the inside of the cell is negatively charged. So there's two opposing forces here, right? Electrostatic pressure pushes the potassium into the cell, where concentration gradient pushes the potassium out of the cell. Because the force of the concentration gradient is stronger, that's the force that's going to win out. So if this channel opens, potassium is going to flow out. So if you're going to take one thing away from all this, just know that at rest, sodium is going to be drawn into the cell if it can move, and potassium is going to be drawn out. And why I'm making you remember that is going to make sense later on. The last thing that I'm going to mention is just a little bit of terminology. First, depolarization. This is a change in the membrane potential towards zero relative to its normal resting potential. So something being depolarized means that it's becoming more positive or less negative, however you would like to think about that. So back to our previous example here, sodium going into the cell, that's a positively charged ion going into a negative compartment. It's going to make the inside of the cell more positive or less negative. So sodium going in is a depolarization event. The next term is hyperpolarization, which is sort of the opposite. A change in the membrane potential away from zero relative to its normal resting potential. So that is going to be making the inside of the cell more negative, less positive, however is more easier for you to think about it. In our example here, potassium leaving the cell is going to be a hyperpolarization event, right? A positively charged ion leaving. So the inside is more negative, something positive is being taken away from the inside, so it's going to leave the resting or the membrane potential as more strongly negative. So that's a hyperpolarization event. Another example would be if chloride, a negatively charged ion, were to enter the cell, it would make it more negative. It's a hyperpolarization event. So yeah, depolarization towards zero relative to negative, so becoming more positive, less negative. Hyperpolarization away from zero, so becoming more negative, less positive. Okay, that is it for our first unit. Next time, I'm going to explain to you why I spent so much time talking about ion movement. I promise it makes sense and it pays off.